In this video, we will study the construction of recombinant DNA. The basis of genetic engineering actually lies in the formation of recombinant DNA. Recombinant DNA means manipulated DNA, in which the gene of interest is inserted, and this is done with the help of plasmid, a vector which is present in E. coli bacteria. When this recombinant DNA is multiplied with the help of plasmid, the gene of interest also amplify, and by following the molecular biology rule, that is central dogma, this DNA will make messenger RNA, and messenger RNA will make recombinant protein. This recombinant protein has all the desired characters. The most important step is the formation or construction of recombinant DNA. This recombinant DNA is formed, that is, the alteration in DNA is done with the help of restriction endonuclease enzymes. Restriction endonuclease enzyme is a group of enzymes which cut the DNA at specific sequence. These specific sequences are known as palindromic sequences. Palindromic sequences are the sequences which can be read same forward as well as backward when the orientation of reading is same. For an example, EcoR1 is the restriction endonuclease enzyme which has palindromic sequence GAATTC in case we read the nucleotide base pairs from 5' to 3' direction. When we read the next strand in the same direction, that is from 5' to 3', because these two strands are complementary, so from 5' to 3' again, the same sequence, that is GAATTC, is present. Restriction endonuclease enzyme will cut the sequence little away from the center, and here between guanine and adenine, restriction endonuclease enzyme recognizes the recognition site. It is present between G and A. When restriction endonuclease enzyme cut the palindromic sequence, the staggered end or the sticky ends are formed. When we separate these two strands, these sticky ends are formed. These sticky ends are overhanging single-stranded stretch of DNA. These are known as sticky ends because they tend to stick with the base pairs which are complementary to these bases. And these palindromic sequence which we cut, it is actually present in the plasmid vector DNA. Now, in this plasmid, in between these sequence, we want to insert our desired gene, and that is why from the donor cell we cut the DNA sequences from the same restriction endonuclease enzyme, so that the same types of sticky ends will be produced, and these ends will have complementary base pairs which are found in these sticky ends. This is actually the DNA which is present in the donor cell. In between, the desired sequence is present, and both the sides the same palindromic sequence which are present in the plasmid vector. The same restriction endonuclease enzyme is used, and that is how the sticky ends which are formed in this gene of interest stretch of DNA. Because these two sticky ends, the nucleotide base pairs are complementary, we can easily join these two sticky ends with the help of enzyme ligase, and the recombinant DNA which is constructed, it has the desired sequence in the center, and on both the sides these sticky ends are joined, and that is how we have constructed recombinant DNA. This recombinant DNA technique is used in making genetically modified organisms, formation of drugs like human insulin to cure genetic disorders by gene therapy, and molecular diagnosis for early detection of diseases like cancer. Thank you.